Since the bacterium was detected in Italy in 2013, the European mandate makes all the state members to share in the territory. And this is the current situation in Europe. We have different areas demarcated under eradication that you see in orange, but other many areas are already in containment, including the south part of Puglia, Corsica, Occitania in France, and also the region of Porto in Portugal and the Balearic Islands. The DRC made a study in which, due to different indicators, they ranked the Xylella as the first pathogen in importance in the economic, social, and environmental aspects. Out of the 20 pathogens and the updates, the current updates, has also revealed that Xylella ranked first as the most important pathogen for Europe, affecting the crops. So, if you evaluate the different economic, environmental, and social indicators, Xylella still is one of the main threats for Europe. How Europe reacted to this pathogen. So, since the first detection in Italy and the consequent detection in France, Spain, Portugal, this pathogen, I think, was one of the most regulated and different legislations were implemented. And the most important thing was that they received feedback from different research projects, and they put in place the change in the law according to what the scientists were revealing, and we started to know about the diseases and the pathogen. So in 2021, many of the main European projects finished. So it was time to decide we needed to move forward, and there are many questions open. So science should continue providing outcomes. Very recently, EFSA requested the panel of plant health to update the scientific opinion on the risks of Xylella fastidiosa. And many of the project partners that are present here are participating in this scientific update. So what we did when we thought about the BeXyl, there were many scientific meetings. We took all the questions that were not answered yet from stakeholders, farmers, nurserymen, managers, legislators. And we also took all the results and the feedback from previous projects. And we tried to answer these research needs by the different objectives that you see there. I'm not going to address here. So we decided to build a consortium, huge consortium. We have 31 partners from 14 different countries. And we have countries where Xylella is present, indigenous in the American continent. So we can take the experience of these countries, of those research groups. And also from Europe, we have all the countries that had Xylella present at that time. We have also countries where Xylella is not present, and the project is coming to the end, the 31st of August. We have also many stakeholders. This is a multi-actor approach project. Many of the entities that are participating as a stakeholder help us to develop the results, and this is quite important. So the project was focused on different work packages that you will see today some of the main results. We try to answer, to make impact on three main aspects. To enhance the capacities to prevent, monitor, and find adequate responses for Xylella fastidiosa. To enhance the capacity to control Xylella in those areas that is already established, and mainly by biological approaches. And another important aspect is to provide scientific-based support for relevant European and associated country plant health policies. So this is quite important in a project like this. So as I told you, we can provide outputs so that the legislators can put in place modifying the current legislation. And now you will see a video that we developed in which we have summarized the main project Thank you. And now we will just start with the different work package. The first one will be the work package one that will be presented by Juan Antonio Navas, that is the coordinator of this work package.
Okay, good morning to everybody. So I'm Juan Navas Cortez, and I'm the pleasure to coordinate World Package 1. And today I will try to give you an overview on the main outcomes that we have reached so far on this topic. So the finding drivers of Xylella fastidiosa epidemics in the EU, including climate change. So in this work package, we have 25 partners, so most of the partners that are in BeXyl are somehow participating in this project, and we are coordinating this work package at CSIC, and with the it's also co-leader by Inroy and Maria Networks. So the main aim of this part of the project is to identify the biological, environmental, and climate-related factors that could determine the development of this pathogen in Europe. So I will subdivide the information in three main topics to how we were able to improve within the knowledge of the host range of the pathogen and the risk. So also to how we think we are better understanding the epidemic drivers under current conditions, but also in future climates, and to finish with some insights into pathogen adaptation. So for the first part, we have different approaches. We have used sentinel plots and botanic gardens to expand the range, or to know the range of this pathogen in real field conditions. But also we combine this information with control inoculations under greenhouse conditions in biosecurity conditions, because we are dealing with quarantine pathogens. And finally, we also perform some field and landscape surveys to complement the previous information. So as you probably know, the sentinel plots, as well as botanic gardens and arboleta, are used or to implemented in the field and with selected plants that were common in all our sentinel plots that were in North America, Central America, South America, in places where Xylella is endemic, but also in different European countries, combining both places in which Xylella is present and other in which Xylella is not present, like in Girona or Córdoba. So these plots allow us to obtain maybe information on new hosts for the bacterium, but also interaction with the vector and with the local environment. In a similar way, but even more diverse in terms of the host, because in the sentinel plots we are focused on crops, but in the botanic gardens and arboleta we expand this to other ornamentals, or not only or go beyond the commercial crops. So this was coordinated by the Botanic Gardens Conservation International, and we 6 botanic gardens in Central and South America were involved in this activity. As a recent product of this activity last week was presented, this outbreaks resource kit that is made to facilitate communication, public engagement, and outreach of biosecurity in plant health in general, but focused in Xylella fastidiosa, and all the materials are available, and you can anyone that is interested can use for their activities. So concerning moving just to field and landscape surveys, during the project we were able to identify new strains or new variants of the bacterium that was not present before. As you probably know, the identification of the ST53 of subspecies Pauca that was before present only in southern Italy, and some identification that's really small, not really relevant, causing problems in southern France, but causing damage was only in southern Italy. Also in this region, that this was the only strain was recently identified to new subspecies and to sequence type associated with almonds and grapes. Also, how disease is spreading or expanding in other countries like Israel, Costa Rica, and Argentina. Moving to the next part of these activities are those related with trying to know the host range of the bacterium in control conditions, as I mentioned, in biosecurity conditions. So we explore different, let's say, priority Mediterranean crops like almond, olive, or grapevines, and how these crops react to European strains of the bacterium. And the main outcome was that these crops differ in susceptibility. That this means that the risk is not uniform, so we depend on the strain, will depend on the host. And together with this, let's say, priority host, was also evaluated other alternative hosts like carob or dragon fruit, because we're planning or trying to obtain the reaction of this crop, because it can be used in places where Xylella was present, and to replace the crops that were affected. Also, we expanded from not only crops, but also in forestry species or other hosts that are member of the landscape, like electrism or sparking function. So we were not focused only on crops, but also in these other Mediterranean hosts. And we could know that these species can also contribute to the pathogen persisting and also to the outbreak pathways apart from the cultivated host. So we also address other outside Europe, tropical, subtropical hosts, mainly in Costa Rica and the US. And we also were able to identify that these hosts living in places where Xylella is endemic, is present, can also support infection. And so have been also considering if we grow these crops in our conditions, and because can also support the infection by the bacterium, and more importantly, that can also host asymptomatic infection. So not solving symptoms but developing. So in summary, so host species are important because different susceptibility even at cultivar levels, also strains, subspecies, but also strains within subspecies can differ, can interact, and we have to consider that not always the bacterium cause symptoms that can be associated to the bacterium. So the host range and the pathogenicity of the different strains has a complex interaction that have to be considered when we address this disease. Moving to the second point, understanding epidemic drivers under current and future conditions, we have evaluated at different levels. So using climatic suitability and soil suitability modeling, control environments, and vector, and how this climate could affect the vector. So we performed a global analysis for the European continent concerning how climate or which areas in the EU could be more suitable for the establishment of the pathogen based on climatic conditions, but also in soil characteristics. And we can see that are almost coincident, but not always. So we can find some areas in Europe that could have very suitable climatic conditions, but not geographic conditions, but also the opposite. But some others in which both factors could allow the establishment of the pathogen. So to go more in deep in these relationships, we have different experiments in control conditions in which we are evaluating the interaction between temperature, elevated CO2 that are the most relevant components associated to climate change, but also water availability and how also the host can interfere. So, and again, we found a complex interaction among all these factors that have to be considered. So we realized that these conditions can affect establishment of the pathogen, but also how severe, if established, can be the disease in certain places. Concerning the vector, we also evaluate the effect of temperature on the development of the vector, and concluding that in particular high temperature can affect survival of the vector, and then therefore how these vectors can persist and transmit the bacteria. So temperature can modify the survival, the seasonal activity, and the potential transmission of this bacterium according to temperature. Moving to the last point, so insights in the pathogen adaptation. So we evaluated how genomics can be used to track introduction from host seeds. So how genetic traits can be linked, for instance, to adaptation, and how we can identify the hiding diversity and evolutionary potential of this bacteria. Just a brief summary of many works using genomics to get some information on to track introductions. For instance, with using genomics, we were able to identify the origin in this particular case in Portugal, but also in other places in Europe, what could be the origin of the infections in a particular place. That can be also used how the bacterium could be adapted from a host sieve. For instance, those strains that were originally specialized in coffee, how when they move infecting grapes, they were more adapted to grapes, but losing their activity in grapes. So how in coffee. So how this population can adapt when arrive into new regions. So also these genetic traits were associated also to adaptation, and how we can identify genetic and physiological traits that can influence activities like the persistence, the movement of the bacterium, also the specificity of the host, and in general, we were able to evaluate the capacity of the bacterium to generate new variation using genomics. And finally, so using this kind of type of approaches, we were able to identify hidden strains of the bacterium using public sequencing data sets, and where some strains that were not previously identified in different countries, but also a potentially new species in Xylella that was identified using this technology. So maybe the genetic diversity, the host range, and the global distribution of the bacterium is broader than we could expect with the current information we have. So we have the need to continue with this genomic surveillance, let's say. To finish, so just some summary or conclusions. So we can say that host range is broader and more complex than expected before. So the outbreak risk depends on local epidemic context. We cannot talk about risk at European level. So we identify environmental conditions that save current and maybe future risk. How genomic tools can develop, how the population moves and adapts. And just to finish, let's say, take-home message, future risk for Xylella fastidiosa cannot be assessed from pathogen presence alone. So depends also on how the different epidemic components, and we think that it's clear that we need to target surveillance, preparedness, and adaptive management in, let's say, current but also future outbreaks of the in Europe. With this, I finish. Thank you for your attention. I take 1 minute more, but it's okay.
So good morning, everyone. Well, I'm Antonio Vicent. I'm from Libya, and I'm together with Pablo Jarco. We are leading working package 2 on new strategies and improved methods for surveillance, early detection, and monitoring of Xylella fastidiosa and its vectors. And I asked my colleague and friend Juan Antonio Navas to help me with 2 of the tasks because they are more related with remote sensing. I'm not an expert in remote sensing, but he's an expert on that. So he will explain these two tasks. So as you can see from the title, the idea behind this working package is to improve all the methods that we have to detect Xylella. And that's very important because it's where the decisions are based. I mean, risk managers, plant health services, and TPOs, they have to take decisions, and for that they need to know where Xylella is present, and it's not only to know where is Xylella present, it's also which subspecies, which sequence type. I mean, there is different level of genetic resolution that are important in terms of, which plants you have to eradicate or which plants you cannot replant in that place. So these things are always based on this method. The outline of the, okay, I think it's a different movie. Blanca. Sorry for that, but
Ya, pero es que va lento. Because we don't really. Okay.
Okay, sorry for that, and thank you very much, Blanca, for helping me. So these are the six, let's say, tasks or the six approaches that we did in this working package to improve both the detection and surveillance methods. First is in relation to the vectors. So, well, I will explain better in the specific slides for this class. Is how to improve the current method for vector monitoring to capture the vectors. Then this is a quite large task. There are many groups involved in that. Well, sorry, because there is also all the groups that are involved in this working package. So you will see some of the logos in the slides. Okay. This obviously is quite important because molecular typing is necessary for the detection of Xylella to characterize, as I said, the subspecies, the subcase type, etc. Also, these are the two tasks that Juan is going to help me to present. One is on the early stage of plant phenotyping of asymptomatic Xylella fastidiosa infections, meaning that sometimes when you do experiments, especially under quarantine conditions, symptoms are not visible or will take a long time to be not visible to the human eye. So there are some image and sensors that can identify some physiological disorders in the plant that are related with Xylella and are not seen by our human eye. So this is what Juan is going to explain. And in fall, we will go up to drones and to airplanes and satellites with the idea to improve the counter surveillance campaigns, also using this satellite data and airplane, etc. 5 will be about the use of dogs, canine olfactory. I will explain this later on. That we think could be quite helpful for border inspections or nursery inspections. This I think is quite interesting. And the last is the only one where I will participate doing work at IDEA is developing and optimizing statistical designs for the surveillance of Xylella fastidiosa and its vectors. So we will go one by one. In relation to the vector monitoring, it's not nice to start with not very good results, but when you do science, sometimes you find good results and relevant things. Other times you find that. Okay. In this case, the standard method, for monitoring the vectors for Xylella is basically sweeping nets or the yellow sticky traps. So the idea behind this task was to use these black light traps in order to see if they can improve the efficiency of the vector monitoring in relation to the yellow sticky traps and the sweeping net. And especially sweeping net is costly in terms of human resources, etc. So basically, after several years of experiments in different locations, these black light traps were not as efficient as the current method. So we cannot recommend this system as a replacement for the standard method that everyone is using at the moment. So as I said, this is not fantastic results, but they are results. Okay, so the work was performed and the experiments are there. In relation to the molecular approaches for quantification, typing, and in situ detection of Xylella fastidiosa in plants and vectors, this is quite challenging for me to explain because I'm not a molecular biologist, so please, Blanca, correct me if I'm saying something wrong. Also because there are several groups involved in this task, so well, I'll do my best. Basically, this is work that was done mostly in Mallorca, and the idea is to test that was a new method at the time when we started with the project, that is the droplet digital PCR, that it was supposed to be an improved method in relation to the qPCRs that were a standard method at the moment. So basically, using this method, it was possible to confirm several mixed infections by different subspecies in Mallorca, Iceland. And this is also important from the epidemiological point of view, because also there may be events of recombination, etc. So it's important to have this level of, let's say, molecular resolution in the This was also tested in this paper that was published, modeling the accuracy of different molecular diagnostic tests. And to me, what is probably the most interesting result is number 3, is development of a high-resolution diagnostic approach based on targeted next-generation sequencing enrichment. That has a very practical, let's say, relevance, because to get to this level of genetic resolution, usually in the past you need to isolate the strain that with fastidiosa, so it's not easy to get in a petri dish a strain of Xylella. So in this case, it's possible to get at subspecies level directly from the plant. Okay, meaning that then the labs and people who are taking decisions, they will have much more information than in the past. So I think this is quite relevant results for risk managers and people who is taking decisions in the field. That's another relevant result, and I think this also you will see if you come to Bari to the conference in a few weeks. This is the monitoring and surveillance campaign that the Blood Health Service in Apulia was doing, and the initial idea was to optimize. You see here there are 2 matrices, almond, grapevine, both from petioles and from cuttings, with the idea to improve how these samples are processed in the lab. But starting to do this work, as I said, with a very practical focus, it was found that new outbreaks of Xylella fastidiosa subspecies fastidiosa, ST1, affecting mainly grapevine, and also multiplex ST26, affecting mainly almond. And you will see that this is one of the field visits that is scheduled for the conference in Bari. And here you see the 3 different methods, LAMP, MPA internal flow, and the droplet digital PCR that were compared in this context of almond, grapevine with petioles and cuttings. Okay. This, again, it has practical let's say, use for the labs and for the diagnostic people, because here you can see the diagnostic sensitivity, both for grapevine, for almond, and with the 2 methods, the C-LAMP and the droplet digital PCR. In general, you see that the performance on almond was better than on grapevine, and also the droplet digital PCR was overall better in terms of sensitivity than the C-LAMP. Okay, so this information is relevant also to calculate sample size, etc. And there was also a group in Costa Rica doing also some improvements to the current molecular methods. They were also testing this flow FLISH diagnostic technique that basically they concluded that it could be useful for endemic context, endemic scenarios, but is not sensitive enough when you are doing detection surveys in disease-free areas. So it's something that could be useful when you already have a disease in your place and maybe you want to monitor how the disease is, in terms of prevalence, etc., but not for detection surveys. And also they were using this real-time LAMP, both in coffee and avocado, and the results in coffee were quite good, but not the same for avocado. So again, it's some information that is useful for our inspections here or there in the field. And all of this was, I would say, merged in this test performance study, because there are, you see, a number of laboratories involved. What we call also ring test is where you put, some techniques. In this case, was the droplet digital PCR, and then the labs are working with a similar set of samples. They report the results in, blind samples, etc. I mean, this is a standard method that is just, and the EPO is one of who established this system of testing the performance of molecular diagnostic methods. Well, here you see, as I said, all the laboratories that were involved in different countries in the EU, well, in Europe, because it's also UK. And here are the results that basically it was a validation of this droplet digital PCR. And Blanca and Xylella local solid, they took a step forward with it because sometimes you get this test performance study. Okay, it's very good. The digital droplet is working very well, but at the end, it should be used by the laboratories, the NPPOs, the Plaza service. So they organized bilateral meeting with EPO. As you know, EPO is providing the standard diagnostic protocols together with the European reference lab. So basically, they communicated all of the results to the panel of EPO, diagnostic for Xylella, with the idea to update the current standards of Xylella protocol for diagnostics. And now, Juan, is your time. Thank you very much for helping me with this. Maybe we'll take a bit more time, but our working group has much more time than the others, so
So you know already that Xylella can show induce symptoms that are not specific and can be confounded by other issues like lack of water or lack of nutrients, or even asymptomatic, that's even more dangerous because the plant is infected and the vectors can transmit the bacteria. So to overcome these limitations, we developed a different level strategy to identify asymptomatic infections. The first one was in ground level. Also, we started in control conditions to identify plant traits that could be associated to Xylella infection. For this, we use this leaf clip, so that measure different components. In one case, just some pigments, but other ones that can measure the full spectral signature of the plants, and also important activity, the porometer and fluorometer that measure how efficient is the plant in the growth the transpiration of the plant, because as you know, when a plant is under stress, tend to close the stomata and this reduce the transpiration of the plant and increase temperature. So using these techniques, both in control conditions, but also in the field, we have identified a series of plant traits that were specifically associated with infection by the pathogen, even in completely asymptomatic infections. So the next level was to develop this phenotyping platform, in which now, instead of just single measurements in the leaf, we are able to scan the whole plant. This is in control conditions in which we have tested so far olive and grapes that were inoculated with the bacterium. And the one big part of this really challenge was to keep only the information that could give us these related traits that could be associated to the infection that we already identify with the other sensors. So in this case, we developed a complex pipeline, but just to obtain real vegetation tissue in which we were able to have the spectral signature. So then you have this full spectrum of the plant that could be also obtained by the other proximal sensors, and we follow the same strategy just to identify or to confirm the plant traits that were previously valid for control conditions or field conditions using leaf clip proximal sensors could be also valid for the whole plant. Then the next level was to move above, so using airplanes or drones. In our case, we use airplanes and the sensors, the same type of sensor, not exactly the same, but that provide the same information, temperature, and the spectral signature were installed in this airplane, and we were able to scan really thousands of hectares in a single flight. We performed in different campaigns, mostly in Mallorca, in almonds, affected by airborne leaf sport, and also in grapes affected by Pierce disease. So we performed different campaigns during the project, obtaining thermal and hyperspectral high resolution images from using this approach. Also, we wanted to test if the information of the models we developed in Europe, in Mallorca, or previously to BeXyl during the Ponte and Mes factors, we were working in a similar approach in Puglia, in olive affected in this region, and also in Alicante. We wanted to know if we go to a new situation like in California, in almonds affected by the same disease, almond leaf spot, if the models or the plant rates that we identified in Europe would be also, let's say, translated to different environmental, cultural conditions, and also the strength of the bacteria. Using the same information, high resolution, hyperspectral, and thermal, we are now, this work is currently being almost we're burning all this result because we have to present a deliverable very soon. So by 1 month time, we will provide all the information on how both the different approaches can be related and how can be practically used to detect either asymptomatic early infections or even in some other cases, just damage that sometimes is relevant to identify where the bacterium is causing really damage. And I think this is enough.
Thank you very much, Juan. And also just to highlight in relation to these two tasks, the first one that this work with the phenotyping means that you need to inoculate plants in a quarantine facility. And that's, really costly in terms of resources, time, etc. And also the remote sensing is also important to at the scale, when you have to do a survey in a very large area and you have a limited number of inspectors and limited number of cars. With these techniques, maybe it's not 100%, but then you can allocate some areas, as Juan was explaining very well, where you can deploy more resources because there is an anomaly or something there, and then you can send inspectors in a case of risk-based approach for the survey. Okay, and in this case, as I introduced before, the use of dogs, maybe you know there are a really famous paper using dogs for the detection of Candidatus liberibacter in citrus in Florida and in California. But in our case, the objective was not to use these dogs for field detection. It was for inspecting nurseries or when you have a container of plants coming into the harbor. And this was done in 2 years by the colleagues from Apulia. And the results, as you can see here, that you can see a very high sensitivity. Even for me, I was not really keen to believe these things, but the values are extremely good. Also, they trained the dogs not only with the plants, also with the soils or the substrate that there was with or without the plants infected by Xylella. And it's true that training the dog is costly, and it's something that is not that easy. But when you have, as I said, thousands and thousands of plants in a nursery or containers, this again could be an assistance to the inspector to focus maybe the sampling in one specific plant that the dog is spotting some smell or something that the dog associates to a Xylella infection. So I hope this work will be published also soon because it's, I think, quite relevant results. And the last part is where I participated a bit is on optimizing the statistical designs because all of these are different methods, plant, aerial methods, the dogs, but at the end you need to come up with a sample size and how you allocate the samples in a nursery or in an area. In this case, we made use, it was our group and the group of INRAE, Samuel Subiran, of using epidemiological models. And what is the advantage of epidemiological models? That we can simulate scenarios based on the data we have. We assume that the models are not perfect, and models are a simplification of reality based on the data and the knowledge you have. If your knowledge is incomplete and your data is incomplete, your model is going to be incomplete, but it's the best that you have. And the idea behind that is that you can simulate different scenarios that you cannot do really in a real situation, because in a real situation you cannot allow the disease to spread. You need to do something. Here, the question is when the risk manager has to take a decision, which kind of survey I have to do, which kind, in terms of maybe the commission to decide if I go for containment or go to eradication. It's good to have some outcomes based on these simulations studies to say, okay, if you follow this road, maybe you don't have an accurate estimate of the number of trees that is going to be removed or that are going to die, but at least you can compare this containment, eradication, this kind of survey, and take a decision based on this relative comparison of the outcomes. So, well, I'm not going to explain this in detail, but basically we use the area in Alicante because, well, I'm from Valencia, so we got all the data from the Planter service, very detailed data, 3 by 3, all the results from the last years. And basically we compare a scenario of no intervention, of basically doing nothing, and then we implemented all this scheme that is in the current regulation with the detection surveys, demarcation, infested zone, buffer zone, and we play with different, for instance, sizes of the buffer zone, different rally for eradication, and very important, different surveillance strategies. You will see in the next slide. And this was compared, evaluated in terms of effectiveness and in terms of efficiency. Meaning effectiveness is how can you reduce the number of infected trees, but you don't want to remove the trees that are not infected. So this is also, let's say, a side effect of eradication, that sometimes you remove trees that are not infected because you are working with rally. So this, we call it efficiency. So meaning we need to find a balance of a strategy that is effective, but also efficient in terms of not removing so many trees that are not infected. Well, this is the paper that is published in open access, all the code, the data, it's available. There are some graphs that I'm not going to explain in detail, but basically you have the red line is the line of doing nothing, so you allow the disease to spread in a theoretical scenario. And these are the different strategies, as I said, with different buffer zone size, different rally for eradication, and very important, different surveillance strategies. So basically, the results say that all outbreak management plans were effective in relative to the baseline scenario of doing nothing. But the main conclusion is that surveillance is key for the performance of outbreak management, meaning that if you don't know where Xylella is present in your area, if you do eradication or containment, you are just, playing in a very incomplete reality. So it's very important to know where the disease is, and that surveillance is key for that. And in the strategies that EFSA proposed, one-step and two-step approach, the two-step approach was clearly much better than the one-step approach. Also implied the use of much higher number of samples, but is the way that you can define where the disease is present. And that's just to conclude, another work that in this case was done by the group of Alexander Subiran. And basically, the strategy here is to make use of the results of your previous survey. So it's like you plan every year based on your results of the previous year in a kind of a Bayesian approach. So I'm not going to go into the details for the questions, but basically with this, you can adapt every year based on your previous results, and the strategy for the new year can be different in different terms, because sometimes you have a kind of aggregation of the disease, there is a spatial connection between the plants, etc. So, well, as I said, I'm not going to go into the mathematical details, but this is a quite realistic approach to improve the samples. And as I said before, for the diagnostic, we were in close contact with EPO, and for this part, we were in close contact with EFSA in two ways. One is the EFSA working group on pest surveillance, that one is one of the members of this working group, and also the working group that already Blanca explained on Xylella fastidiosa risk assessment, that we have to deliver before June, now it's in September, and you will see some of these outcomes much more improved because we did also some additional modeling work in the framework of the working group with different simulations and different potential scenarios. And with that, I finished. Thank you very much. I'm sorry if I took a bit over.
Another important point for avoiding the spread of Xylella fastidiosa is the movement of plants that are Xylella free, so the safe movement of plants. For that, we have here a very important stakeholder participating in the project, is the European Nursery Association. So one of the main requests from this association was what can we do to help nurserymen to work, especially in the areas where Xylella is detected, it's completely forbidden to move the plants from that areas. So the work package 3 was dedicated to work on the use of thermal treatments, because the only information we had before was a few reports for grapevines that were adapted from viruses work. So we try to move forward analyzing the how thermal treatments could be used in different plant species. And for that, we have developed a video, so I think it will be much easier for you.
This short video aims to illustrate the activities and results of the BeXyl project concerning the thermal treatments for the production of Xylella fastidiosa-free nursery materials. But why do we speak of propagation materials? Xylella fastidiosa lives only in the wood and is therefore transmitted and spread efficiently at long distances with vegetative plant propagation materials such as whole plants and parts of plants. Furthermore, due to its high genetic variability, Xylella can cause serious epidemic diseases in a huge number of important crops, ornamentals, and species of the natural flora. Today, it represents a major threat for the global plant trade and a true nightmare for European nurseries. The idea for a specific work package in BeXyl came from the previous Horizon projects, Ponte and XF actors. During their final meetings, nursery representatives explicitly required EU authorities to provide higher attention, research, and specific technical solutions to help the sector, which was affected by growing negative commercial impacts and damages. The proposed solution is represented by thermal treatments. This consists of the application of high or low temperatures using water, air, or vapor, capable of killing pests or pathogens outside or inside fruits and plant propagation materials. It is a physical, economically sustainable, and environmentally friendly system. However, it requires studies and validation of specific protocols and conditions of treatment, meaning exact times and temperatures, able to clean the nursery material without affecting its quality and vitality. Despite the existence of dated scientific literature and official standards for hot water treatment against some harmful organisms on a few host plants, like grapevine, pome fruits, and strawberry, this broad-spectrum phytosanitary tool remains largely underutilized. It requires further study to be applied against pathogens like Xylella and a higher number of important plant species. Besides nursery production, thermal treatments can find a wider application for the international exchange of materials for scientific uses and quarantine purposes. The research, conducted by a broad partnership including research institutions, nurseries, thermotherapy, equipment manufacturers, and 2 nursery associations, has been organized into 4 different tasks. First, to verify the efficacy of temperature and time combinations on the bacterium in vitro and in infected tissues. Second, to verify the effects on the quality and viability of plant materials without the bacterium. Third, to validate the best thermotherapy protocols on healthy material, at a medium scale in nurseries. And fourth, to evaluate operational treatment conditions and alternative technical solutions. In vitro, using up to X strains of 3 different subspecies representative of the bacterium's genetic variability, as well as advanced fluorometric and molecular techniques, we evaluated the effect of X temperatures. These range from -15 up to 50 degrees Celsius, tested over different times, from a few minutes up to several days and weeks. We evaluated their impact on biofilm production, multiplication, and viability of the bacterium. Regarding the trials with hot air and hot water against the bacterium in vivo within the host tissues, we faced great difficulties with artificial infection. We used both potted almond plants, inoculated by microinjection, and cuttings of ornamental shrubs from naturally infected plants in the field. We successfully identified grapevine, naturally infected by the strain ST1, as the best model combination. Thermotherapy trials conducted with healthy plant material, whole plants for planting, dormant buds for grafting and cuttings, were carried out on a small and medium scale in laboratories and nurseries. We used different representative varieties of many host species: olive, grapevine, almond, cherry, peach, walnut, fig, white mulberry, oleander, lavanda, polygala, laurus, acer, elm, 5 quercus species, and others. As expected, although there are notable differences between species and varieties, dormant deciduous plants better tolerate heat treatments at high temperatures. For some other evergreen species like olive, even if the maximum conditions tolerated seems unable to eliminate Xylella, the treatment was verified useful to control other pathogens, such as Pseudomonas sevastani. The olive and almond trees HW treated in 2023 were checked for microbiome modifications. Then, in 2025, planted in open field in Puglia for phenological observations and the detection of potential epigenetic modifications. To evaluate the viability and multiplication capacity of the treated plant materials, traditional techniques such as rooting percentage or grafting success rate were compared with new and faster methods. These include stereomicroscopic observation of the tissues, budding in water, and an electrical conductivity test called the electrolyte leakage test. The trials, carried out mainly in Italy and Spain over 4 years, demonstrate that several important variables play a crucial role: the harvesting time, the level of dormancy, the diameter of the propagating material, its nutritional status, as well as pre- and post-treatment conditions. These must be further investigated with a larger panel of species and varieties. Similarly, some limited, very preliminary experiments with cold treatments and hot vapor could also be promising for future nursery applications. The most tangible result achieved within the project is certainly a draft EPO standard for the control of Xylella with hot water thermotherapy for grapevine, fig, and white mulberry. However, the large amount of new knowledge acquired through a huge number of experiments, including negative ones, requires continuation of the studies. This gives hope for expanding the standards to a greater number of species hosting Xylella and for using heat treatments against more target organisms. Finally, some important side activities confirm the interest and positive perspectives to extend this research line, ranking the nursery importance of Xylella host plants, classifying them by technical and physiological characteristics, the preliminary results of a questionnaire for nurseries, and the success of divulgative seminars, all demonstrate a strong interest in the future of these phytosanitary tools. Let's work together to get a sustainable and healthy future for plants and nurseries.
Now, we are a little bit late with the program, but we have time for questions because otherwise if we wait till the end, it will be a lot of information. So if you want some clarification or question, just go ahead.
Thank you. I have 2 questions, very easy. I am working on plant health for those who don't know in Southeast Asia, so I am very sort of looking into the future. On the a question on the simulation based on epidemiological models, you consider also the vector dynamics, I assume, in the epidemiological model?
Yes, in this particular model, there is one parameter that considers the transmission efficiency, meaning that the vector is playing there. And we implemented 3 levels of reduction, like 90% of transmission, 50%, and 10%. Meaning that this simulates like you are doing spraying for vector control or you are removing inoculum because then you have less number of infected vectors. The only thing is this again simulation, so we don't have clear information to say how many sprays will be translated in this particular number of 0.1, 0.5, or 0.9. But basically, when we did a sensitivity analysis of the model, that was one of the most influential parameters in the model. So meaning that vector control is fundamental for avoiding exploit. And I think this is in line. You have Gianni in front of you. I think this also the conclusion of your previous papers on that matter. Yeah.
Thank you. And the other question, I mean, very interesting, the issue of sniffer dogs.
Oh.
Do you know if someone has got on rats because there are rats being used to detect tuberculosis, detect the demining things, so and they're easier to train than dogs.
Yeah. So well, I'm not aware of that. Honestly, but the software maybe will be exploring another project because, as I said, for the dogs it's quite expensive to train them. So that's to me probably the main limitation of this method that apparently it works very well, but you know all the time that you need to train the dog and all the setting you need with Xylella, without Xylella and things. So yeah, but I wasn't aware of this or the option. So this work was done, correct me Blanca if I'm wrong, with the Association of Dogs in Italy or Animal Welfare or something. So there is also not only the plant pathology scientist, also association in relation to dogs.
And are these dogs now working with BeXyl?
Well, I think that in practice they are maybe not implementing that because I'm not sure if this is allowed currently. I mean, if they are using the dogs already in a pool. I don't think so. I think that was in an experimental setting. And I don't know, it's something maybe to explore if, because I guess they have to follow the official procedures for sampling, etc. And if dogs are not there, maybe they are not using that, but it's something that can be done in the future, I guess.
The idea is where to use the dogs maybe at the nursery level or at the point of entry. Yeah, because with the fields they can be very confused. Thank you.
I have a question. Thank you for the very interesting presentation and the really nice results. Regarding the early detection of asymptomatic plants, will this be a protocol soon, or can this be implemented then for member states to do the surveillance? Or,
Yes, in particular, well, at different levels. Maybe in nurseries or even for field surveillance, can help inspector to select the right plant. Because in particular in areas where Xylella is not present, not known to be present, it's really a challenge for the inspector to select the plant to be sampled and tested in the lab. So the leaf clip, we already have very robots or that are working on different situations in olive, in almonds, and grapes. And we tested both in control conditions and in field conditions. And we have certain traits that are common to the different pathosystems, let's say, but also some that are related to a particular host. And also we tested against another vascular pathogens, the Brazilian valley, Brazilian leaf, that cause similar symptoms, or because both growth into the vascular system. And we found also traits that are related to the two parts, one of the pathogens. So this is one news. And this is really important connecting with surveillance, because now for Xylella and other priority pests, it's compulsory to use a statistically based approach for some surveillance design. And how efficient is the inspector selecting the right plants are component together with the diagnostic sensitivity, and this affects the mathematical examples are example. So this can help to the inspector, but also in the nurseries, also can be used directly by the nurserymen to test their plants. The next levels are most, let's say, need a farmer association, because you have to deploy a flight, so with all sensors, the process of the images is not easy. So a different level can be used, and now we are testing how we can combine this information coming from satellites. The problem of satellites is that we, for in particular for early detection, we need high spatial resolution and high spectral resolution. And to have both in one satellite is currently is not possible. So you have either high spectral resolution or high spatial resolution. If you have a resolution of kilometers, it's not useful for even for trees. But if you have a very precise, very low spatial or high spatial resolution, but low spectral resolution, you could miss. We in the spectral signature, we identify certain regions, not the whole spectrum that is associated with the symptoms, not symptoms, infection by the Xylella in the plants, and you will miss this information. So at different levels, this technology is ready to use. The only thing may be to a company that implement in a tool the information coming from the sensors, so the proximal sensors, in order to be processed directly in the tool. This would be the next step. And the airborne or satellites is a different level, maybe a regional level, or even at a question of the European Commission, let's say, to probably put in practice this. But the technology is already ready to use.
More questions? So we have now a coffee break. Since we are a little bit late, maybe we can go have the coffee and then come back with the coffee. So.
Let's try.
Can you hear me now?
Yes, now I can hear
Okay.
you. Great.
Okay, perfect. So go ahead, you can present yourself, okay? Okay?
Yeah, just let me know first if you can see my presentation.
Yes, in full mode. Okay.
Great. Okay, so let's get started. Hello, everyone. My name is Ophir Bahar. I'm from the Agricultural Research Organization in Israel, also called the Vulcani Institute. And today I'll be presenting some of our work and results that is related to work package 4, dealing with developing genomic tools linked to Xylella fastidiosa resistance and their application. So in this work package, we have 14 partners from different countries and institutions in Europe. We have partners from the United States, Central America, South America, and Israel. A very quick background, because I imagine most of you are familiar with this. Xylella fastidiosa is a bacterial pathogen threatening multiple European and Mediterranean crops. It's spread through insect vectors, and once trees are infected, there's no reliable and effective cure to make the tree healthy again. The main impact at present, at least, is on olives, almonds, and grapevines. What you see here in this illustration, or in this picture on the left-hand side, you see a transmission electron microscopy image showing the vascular tissue or the xylem vessels, the water conducting elements in the plants that are packed with bacterial cells, basically blocking the water flow and leading to the desiccation symptoms that we are used to see on infected plants. And on the right-hand side, you see the Philenus pomarius, the main vector of Xylella in Europe. So how do we manage Xylella fastidiosa diseases today? So there are basically a few main approaches. The first one is vector control, which is done either by spraying directly the vector or by managing ground vegetation where the vector completes its life cycle. We use sanitation, so we remove infected trees and different quarantine measures to restrict the movement of infected material. We use, or we should use, healthy and certified plant propagation materials, so we're not spreading infected plants from nurseries. And these three approaches are effective to some extent, but they are costly, and they're eventually only temporal solutions. So we have to keep applying them time after time in order to keep the infection in check. Now, plant resistance, on the other hand, can be very effective and a very durable solution. When we say resistant plants, we mean plants that can restrict the multiplication and the movement and spread of the bacteria inside the plant. The result of that is that we have trees that are less affected by the pathogen, so they appear healthier, their yield is less affected by the infection, and also very important, these trees do not serve as a good host for vectors to acquire the bacterium, because the bacteria are in very low concentration and they're not widely spread in these trees. So we also gain by that reduce epidemic spread. So there are a lot of advantages for using resistant plants. Some examples for resistant plants to Xylella fastidiosa, I'm sure most of you are familiar, are these two genotypes, olive genotypes, Lecino and FS17, that are resistant to Xylella fastidiosa subspecies pauca. You can see on the left-hand side an example of a resistant tree compared with a susceptible tree on the right-hand side. So what is it that we still don't know about resistance in relevant crops for the EU and the Mediterranean? So as I just mentioned, for olives, there are basically, well, at least when this project started, there were only two genotypes that were known to be resistant to Xylella fastidiosa in olive, and these are the two genotypes I just mentioned. And in almond, there was no proven sources of resistance in Europe at all. Furthermore, there weren't any genetic markers to be used for breeding programs to breed for resistance, and that's a major obstacle in breeding when you don't have genetic markers. And furthermore, we have a very partial understanding of how does resistance to Xylella fastidiosa actually work. What are the genes that are important for this phenotype, and how exactly the bacterium is restricted in the plant. So our mission is to expand the arsenal of resistant sources, and the primary objective was to identify new sources of resistance in olive and almonds and in other important crops. The key approach, as I will show you very soon, was to screen a diverse germplasm collection across Europe and try to identify specific genotypes that are resistant to Xylella fastidiosa. The end goal would be to find new resistant varieties to provide genetic tools for faster breeding, and a better understanding of how resistance works so we can generate durable resistance. So we're taking a four-pronged approach. The first one, as you can see in this illustration, is the screening of large collections of genotypes of primarily olive and almond, generating new resistant breeding lines, then developing genetic markers for resistance, and as I mentioned before, understanding the molecular basis of plant response or plant resistant to Xylella fastidiosa infection. Okay, so let's start with the first aim, and that's to identify resistant varieties. As I mentioned before, the approach here was to screen large collections of genotypes. This has been done in multiple countries in Europe and also in South America, Central America, and in the Middle East. Some of the screens were done, as you can see here, outdoors, so open to natural infection by Xylella fastidiosa, and some of the screens were done indoors under controlled infection conditions. And overall, we've screened hundreds of different genotypes and cultivars from olive and almonds. Well, the key result, we have identified several new resistant cultivars in olive beyond the ones that we already knew. And in almond, we have discovered also multiple resistant germplasm, both in Spain and in Israel. And the impact of these results are that we are expanding our genetic base for resistance breeding, which can be very helpful later on, as I will show you. The next step was to build the next generation of resistant crops. And basically what we are doing here is crossing resistant olive cultivars, so cultivars that are resistant to Xylella, together with other commercial cultivars that have desired agronomical and commercial traits. We generate a large population of seedlings, and these seedlings are later planted in the field and tested for Xylella fastidiosa and other traits. So the key results here, we have generated, as I mentioned, large seedling population that are now under evaluation. And the evaluation goes both ways. One, they are tested for resistance to Xylella fastidiosa to make sure that the resistant trait from the parent is retained in the seedling or in the progeny, and also that they have the correct or the desired agronomic traits for commercial use. And overall, the goal here is to combine disease resistance with agronomic performance and commercial quality. So these kind of population are currently under investigation. And what you have to remember here, since we're talking about trees, the whole process of making these crosses, generating the seedlings, and then also evaluating properties like yield can take a few years, and it's not a process that can be done in a few months or even not 1 or 2 years. Tools for faster breeding. The next step is to develop genetic markers for resistance. So what we can do here is basically, once we have identified genotypes that are resistant to Xylella fastidiosa, we want to know what type of genetic marks they have in their genomes that are associated with the resistance. This is a very important tool for breeders, because when you have a reliable genetic marker, you can try to see whether this marker is present or absent in a specific germplasm. And by that, you have a very good idea whether it would be resistant to Xylella infection or not. And this is much quicker and cheaper than to actually put the plant in the field and to expose it to infection and wait a few years until you see whether it's resistant or not. So genetic markers are an excellent tool to expedite and to make the breeding process much more efficient. The target crops here for developing these genetic markers are olive and almonds, and the development of these markers is advancing and is still under work. As I mentioned, the future impact of this would be that it could significantly accelerate breeding programs for developing new resistant cultivars. The final part is to better understand how plants fight against Xylella fastidiosa. So as we said, we know that there are plants that are resistant to Xylella, like I mentioned, lechino or FS17. They can restrict pathogen multiplication and pathogen spread, but how exactly do they do that? So the approach here was to sequence the genome of two olive genotypes, one resistant and one susceptible. And by obtaining this information of the whole genomic DNA of these cultivars, we can make different comparisons between a resistant and a susceptible genotype and try to identify what are the genetic elements that are present in the resistant one and absent in the susceptible one that are responsible for the resistance trait that is so desirable. So we have completed the genome sequence of two different genotypes, as I mentioned before, Lecino, the resistant one, and Ogliarola, the susceptible one. So the genomes have been completed and now can be used by the scientific community. We have done transcriptomic analysis. So in this analysis, we are looking at what specific genes are responding to infection in a resistant cultivar and in a susceptible cultivar. And by doing this analysis and comparison, we can identify specific genes that are responding to infection in a resistant cultivar and not in a susceptible one, and identify good candidates that can be related to the disease resistant phenotype. And this is also tightly associated to the genetic marker development that I mentioned before, because if we know which genes are responsible for the resistant phenotype, we can also develop effective markers based on these genes. We can also scan the genome and look for immune receptors that are based on structure similarity or sequence similarity based on other more model crops, and develop different laboratory tools to screen these different plants and to screen the function of different genes when it comes to resistance in a more small and rapid scale rather than doing it outdoors, as I mentioned, in a process that can take years. And finally, we're also developing protocols for transformation of olive trees and regeneration, so we can make olive seedlings either expressing specific genes or that can be used to test the function of different genes and to see their importance for resistance. So to summarize this, our key result, we have identified new resistant germplasm in both olive and in almonds. We have breeding populations that are now being established and are under screening for both Xylella resistance and for agronomic traits. Genomic markers are under development in these two crops. And we have excellent tools now to better understand the molecular mechanisms behind resistance to Xylella fastidiosa in laboratory conditions. And the impact of all this is basically expanding our toolbox and options to generate durable resistance to Xylella fastidiosa in important crops for the EU and other states. What is left to be done still? So the immediate priorities of this work package is to complete the screening of the breeding population, to identify the best candidates, to finalize the genetic marker selection and validation, and to validate different immune receptors that were identified through the genome sequencing. Again, as I mentioned, this would expand the options in our toolbox to generate durable Xylella fastidiosa resistant and management for the long term. And hopefully we'll be able to release new resistant varieties to European and Mediterranean growers and provide breeders with better tools for breeding Xylella fastidiosa resistance into these important crops by, as I mentioned, generating new genetic markers and establish eventually sustainable resistance across European crops. And this is it. Thank you very much for your attention.
Thank you. Thank you, Philip. You stay here in case there are questions.
Of course. Mm-hmm.
Now it's time for work package 5.
Yeah.
Yes. So, good morning. I'm Domenico Bosco from the University of Torino, and I am an agricultural epidemiologist. Of course, involved in this work package 5, which deals with new solutions for effective control of Xylella fastidiosa vectors. You know that Xylella, under natural conditions in the field, is only spread transmitted by insects. So targeting the insect is very important to hamper the spread of this pathogen. I will use this.
Or the mouse. Maybe you can do.
No, now it works. Okay. These are the research unit participants from Spain, from Italy, from Austria, and also from Brazil. Just a very fast summary of the work carried out that is structured in 5 different tasks. The one is devoted to the development of entomopathogenic fungi against the insect vectors as biocontrol agents. The second one has been devoted to the study of the viruses associated with this, with the main insect vector, Philemus fumarius, that have never been studied before. We now identify a number of viruses, but it's still a long way to evaluate the impact of these viruses on the insect fitness, but we are doing that. And the third task has been devoted to the study of parasitoids of Philemus fumarius. We identify one egg parasitoid which is relatively efficient, not very efficient, but can contribute to biocontrol of this insect, while the adult parasitoid Veralia auta show always low parasitization rates. It's a very interesting insect, but probably not for effective biocontrol. Then the fourth task is devoted to this to develop agronomic techniques that manipulate ground cover management in these perennial crops potentially affected or affected by Xylella fastidiosa. Why ground cover management? For those that are not familiar with these insect vectors, the spittlebugs, they develop as young stages, immature, on herbaceous vegetations. As soon as they emerge as adults, they tend to colonize canopies of woody hosts such as olive, almond, grapevine, and so on. So when considering Xylella epidemiology in perennial crops, we have to consider the herbaceous ground cover, which is very important. If we target nymphs, we can work on ground cover management. And then the last task was devoted to the study the feeding behavior of Philemus fumarius related to the transmission of Xylella fastidiosa and how we can partly interfere with that. It's very challenging. Then the main outputs, for the main outputs, I will focus only on 2 tasks that are the ones that provided the most advanced results that can be translated to some extent into practice. The first one is the development of what we call EPF, entomopathogenic fungi, as biocontrol agent. Here, just to remind that these are trials conducted in Spain, demonstrated that the blue part of the bar represent mortality induced in the 2 main vectors, Philemus and Neophilenus. So this Metarhizium strain can be pretty effective also against the adults. And going to the field where things are much more difficult, applying this same strain in 3 consecutive years, we can see that the level of these insects decrease substantially, although of course it's not a chemical insecticide that potentially kill almost all the insects, but can be. And also in these other trials conducted in Spain on Neophylenus campestris, in dashed lines you can see the trend of adults and the trend of nymphs in control plots, and you can see that in solid lines the trends in plots treated with these entomopathogenic fungi. The same we did in Italy, and we identify in the lab all these strains of Metarhizium belonging to a couple of different species that can reduce, that against nymphs can cause mortality exceeding 90% under lab conditions. When we went to the field in Apulia, we perform trials including our Metarhizium, the product of reference which is on the market, Naturalis, control, or mowing, simply mowing. Results are not always very clear to interpret, but what we say, okay, here is the pre-treatment situation in terms of nymphs, then is the post-treatment. We can see that mulching of the herbs is still probably the most effective tool, but also Metarhizium significantly decrease the nymph population. In another site of Salento area, results are similar. Here is pre-treatment, here is post 7 days and 2 weeks post-treatments, in which mulching is always very effective, but the fungi are pretty effective. We are not looking for the best option, better than the ones that are available, but we are looking for new tools that can be integrated with the ones that are already available. And for this, we are pretty happy with these results. Plus, we studied the property of these fungi to colonize in plant tissues when sprayed to the plant or eventually to the soil where the plant grows. Some of these strains, not all, have the property to become endophytic, to colonize the leaf tissues, and in this way their persistence in the environment is extended. The point is that once they are endophytic, they change the metabolism of the plant, the taste of the plant for the vector, but they are not severely pathogenic to the insect. They can induce some mortality in some, but of course not like as a direct effect when we target directly the insect. In any case, in Spain, with the Metarhizium bruneum strain, they show some capability of this fungus to colonize both herbaceous plants, but also olive tissues, and also to induce some toxic effect against the insect. In Italy, with our strain of Metarhizium anisoplie, we found a good capability in colonizing tissues of herbaceous plants, although as endophyte, we fail to identify through mortality effect. But in the end, we concluded that all the trials conducted in Spain and in Italy are encouraging and suggest that entomopathogenic fungi can be implemented in an integrated pest management program. We think so. Then the other approach was the one of the manipulation of the herbaceous ground cover. Of course, always against the insect vector nymph. In this way, if we target the nymphs, we prevent the emergence of the adults that are responsible for the spread of the disease. So at the XIC in Madrid, they started with the no choice test, rearing of Philenus under glasshouse conditions on plants that are possibly toxic, and they identify some plants that induce very high mortality in the insect compared to the good suitable plant. Then they moved to the field in several sites under different environmental conditions, from Mallorca to Alicante to Madrid to Galicia, manipulating the ground cover management of vineyards and also olive orchards with 3 candidate species: Antriscus diplotaxis sinapis, according to randomized plot design, and also comparing with tillage. The results are pretty interesting because, just to summarize very briefly, the ground cover dominated by Antriscus cerefolium hosted a much lower number of vector nymphs. And when considering also Sinapis alba, Sinapis was the best repellent toxic plant for Philomus. Similar trials have been conducted in Apulia, and where they use a slightly different approach. They compare the undisturbed natural vegetation, like XIC did in Spain, with mowing the herbs, or sowing, or single species, or mix of plant species, and compare with plugging, which is the measure which is currently applied to destroy plants in order to suppress the insect population. Although under field condition, the results are sometimes a bit difficult to interpret, it was clear that some, especially sowing some mixes, there was a clear suppression of the nymph population, especially if these plants, after sowing, were coupled with mowing. We are therefore proposing some tools for a more advanced vector management, which is now based on plugging in the spring to destroy the plant. We think that on the long run, this is not very sustainable in under Mediterranean conditions. Such a measure implies a loss of water, which is a precious impact on the microbiome of the soil. So we suggest the possibility of a spring control against the nymphs using fungi, so preventing the development of adults that transmit, but also fungi can also be applied in autumn when the females return to the herbaceous cover to lay eggs. And We didn't observe a spectacular effect in terms of reducing prolificity, but we had significant results in terms of survival of the female while laying eggs. If they survive for a shorter period, they definitely lay less eggs. So this is another approach. So there are two key time windows for vector control in spring and autumn. And also, we would like to suggest implementation in the IPM of sowing with selected plant species that are or repellent or unsuitable for spittlebug development, replacing or plugging or the natural undisturbed vegetation, which is not suppressive of the insect. Finally, this is the last slide. The three measures that can be implemented are sowing between the rows of the perennial crop, EPF applications in spring or in autumn, and finally mulching. According to the situation, these three measures can be integrated into integrated pest management strategies schemes of different complexity. We know that according to the different farming structure, one or even all the three measures can be implemented, and this is the final message. Thank you.
Well, good morning. I would like to explain you the advances in new solutions for especially for increasing plant resilience to infection by Xylella fastidiosa. This means that we want to prevent infection by, for example, priming the plant to be prepared to be attacked by the pathogen, or more ambitious is to cure or at least to decrease the damage on the plant by developing new tools. I am the leader of this work package together with Stefan Tomber from the Austrian Institute of Technology. You have the different partners here. During my presentation, I will not show details on what did every partner. I will concentrate more on the description of the results. But I would like to introduce first what we mean about new solutions. Probably you know there is an EFSA report about the strategies based on treatments with products to make a certain control of the disease. This review is based mainly on data from the United States done with the Pierce disease in grapevine, which is a very old disease, very well known. And also some experiments performed in Europe. So the conclusion of the work is that only antibiotics provide a reasonable protection with many other side effects. And since we know that antibiotics are not authorized in Europe, we need to develop novel solutions. That's the idea of this work package. An important aspect is that the idea of the work package is to provide proof of concept for these new solutions, and the testing is in part done in work package 7. Okay, but I have to tell you that it's not sufficient with the duration of the project. So we need much more years to develop these new solutions. One of the first approach was to develop synthetic communities, microbial communities, especially bacterial communities, from bacteria isolated from the xylem, which is the habitat of Xylella fastidiosa. So it has been performed by the AIS in Cordoba, and you see here a tremendous amount of samples from different places, from the xylem of olive trees, even from the vector. And they did an approach which now is very popular, which is we call metagenomics, which is to find the community of the microbiome, microbiota present on the material. I'm not going into details. The question is that it involves next generation sequencing, special bioinformatic processing, and so. But from our point of view, this is very interesting, but do not provide by itself a strain which is of our bacteria, which is necessary or important to make biocontrol. So this is complementary to a second approach, which is the isolation of bacteria from the material. And you can see here the process which is convergent, starting with near 300 bacterial strains, and then evaluating this big collection by testing in vitro different properties, with the idea to select only those strains that have properties that are useful as antagonists against Xylella fastidiosa. I'm not going to detail, but one part also important is the compatibility assays, because we want to make a consortium of different strains, but they should be compatible. For example, it's very frequent that you combine a bacillus strain with a pseudomonas strain, and they kill themselves. So it's important to see the compatibility to finally have a consortium which is really promising. And then in the last stage, the synthetic consortium design. So you have here all these bacteria that have grown in agar plates, in different media, in different conditions. Let us say that some of these strains were discarded because they have some concerns by the safety point of view, and only the ones that have a good promising conditions, but they are safe. Compatibility assays is very important. They did several combinations between the lead strains at the end of the process of selection. And as a brief summary, they developed two consortia. One with a strain of Bacillus aphensis, a strain of Novosfingobium floreni, and a strain of Variovorax. Both, all of them compatible. And then a Rhizobium and Metylobacterium in Novosfingobium strains. Okay, so five strains, combined into two consortia. Another approach to find novel solutions was to test antimicrobial glasses and ceramics. Let me say that, in this laboratory, they are specialized in developing this kind of compounds oriented to other applications, not in biotechnology, but mainly in other processes. The summary is that after in vitro testing, the conclusion in vitro and testing the toxicity in a plant, they saw that there are toxicity in the cases of concentrations, high concentrations, which were reactive. So in this case, we are not sure that at the moment these novel solutions may have a good perspective for the future. Another one, which is more promising, is to test components extracted from the alberujo. Alberujo is one of the components of the olive oil extraction from olive. Okay, and tested several components by different extraction methods, and tested against 3 subspecies of Xylella, some strains from Europe. And the conclusion here was that hydroxytyrosol, which is one of the components, is the most effective. And it's a promising component, which is very interesting on the basis of the minimum inhibitory concentration. There were other components that were interesting. In another case, we tested my university in a collaboration with a company, botanical extract similar to the alberujo, but in this case is the eucalyptus globulus oil. So this component is a mixture of different components. The most abundant is thineol, and is a product which is very popular as it's a repellent, even used in other applications. But we found that surprisingly this product is lytic against Xylella fastidiosa. We tested 3 strains, and the in vitro study gave idea to proceed with implanta test. And these are the results with 2 subspecies of Xylella about disease severity in almond. We have been testing in my lab. Important thing is that in our proof of concept, we use always endotherapy. So it's not spraying the plants, but it's injecting the product in the vascular system of the plant. So you can see here the results. It's clear that it is effective compared with a non-treated control as a preventive application. So that's a proof of concept. The company has been testing this product in Mallorca and I think in other places of the world. The problem for testing against Xylella fastidiosa in the field is mainly the high variability from tree to tree. And this is very important to have in mind. Most of the publications you can find in the journals, I cannot believe the results because we have a strong variability even in plants that are clones made in the greenhouse by endotherapy, and then when you go to the field, you find strong variability, and many times you don't find statistically significant differences. So that's one of the problems. The company is still doing that because they have a patent of this product, not for Xylella, but they are still working on that. This is a way of transferring our knowledge. So it's a botanical extract. The second approach in this part of natural and synthetic compounds is the work with synthetic peptides. And this is our main field of research of my group. And we paid attention to 4 properties of peptides. One is the bactericidal activity. You can see here, Xylella, this is the treatment, and this is non-treated. You can see here how these lytic peptides act. And this is transmission electron microscopy. You can see the action of the peptides making pores in the membrane and producing this burst of the cell. So this is very important because they have direct action against the bacteria. We have several papers published on that property. Another interest is to find peptides which are anti-biofilm peptides, because Xylella is colonizing the xylem, the system, mainly producing biofilm and clogging the vascular system, and then the plant is really submitted to a difficulty of evapotranspiration. And the symptoms are mainly due to that. But also, another important aspect is to check the peptides for plant defense induction. And this is done by doing transcriptomics. This case is RNA-seq. You can see here a plant treated with a peptide and non-treated plant. You see really a tremendous change in the transcriptomic profile in the plant, especially oriented to defense genes. Not all the genes are affected. Okay, and the last one is important, is the phytotoxicity. So peptides that are phytotoxic are not good promises. So from these platforms, we did the implant tests. As first was presented, it's important to work under quarantine greenhouse, because Xylella fastidiosa is a quarantine pathogen of the EU. And we are in an area of Spain where there is no present disease. So this greenhouse is a level 2+ greenhouse. Compared to other places, we have strong restrictions to work with that. Okay, so we inoculate the plants. You can see here one of the researchers working. This is almond plants. This is the size of our plants. We cannot work with big trees. Okay, and inside the greenhouse there is a laboratory, which is also a negative pressure, and that's how feels the conditions of containment. This is the device we use for injection. Is a micro injector. Micro means that we inject 10 microliters per plant every injection. Just in the we try to inject in the xylem, but well, in almond is relatively easy because the xylem is in the central part of the shoot, but sometimes part of the product goes to the phloem. But in any case, it's going to the vascular system. You can see here how we did this. And this is important because it's a proof of concept. Sometimes people say, well, what to do in the field? No endotherapy is the solution, but for us is a platform to testing products and to see, to select the candidates. We did this in Prunus dulcis in the cultivar Abidjan, which is susceptible. Well, Ophir said is resistant, but using the definition that Ophir put in the presentation. For us, you can see here that it's susceptible, less susceptible than other cultivars. So for us, it's a good system, but the problem is that if you inoculate the bacteria, you need 6 months on average to see symptoms and to monitor the bacteria in the plant. For olive trees, nobody tell that, but maybe more than 1 year. So imagine what happens. You start an experiment and you need 1 year to see the results. So we developed a novel system which is using Nicotiana bentamiana as a surrogate host. And Nicotiana is susceptible to Xylella. There is a recent paper from us appearing in in phytopathology using Nicotiana bentamiana to study the aggressiveness of strains from Europe, especially from Spain. Okay? So Nicotiana is a good model for testing products. And you can see here how our peptide BP178 is detected until 60 days, 2 months. So it means that the peptide is degraded, but you can see the peptide in the vascular system of the plant. Another effect, this is for aldone, is that this peptide is cleaning literally the vascular system of the plant. Compare it, for example, to controls, you can see here, which are really blocked by the biofilm of Xylella. And finally, the population of the bacteria decreases if you perform a protective and curative approach, one treatment before 1 or 2 days before inoculation and 1 or 2 days after inoculation. So this is really reducing the population of the bacteria. Look at the variability I told to you. These are cloned plants obtained from in vitro by a nursery, and we have this variability. Okay, but this is working. These are the sequences of the peptides. Sorry, because probably if you are not biochemists, you cannot imagine that. I tell you that K is lysine, is one amino acid. This is a 1-letter code. Yeah, I'm just finishing. And these are the best peptides from all we test. Here there are lytic peptides. There are peptides stimulating defenses on the plant. And there are peptides that are both activities. For example, the first one is lytic on Xylella and stimulates defenses on almond and other plants. Finally, the third approach is the study of bacteriophages that are lytic to Xylella. This work has been mainly performed by EMEA, and you can see here pictures from different lytic bacteriophages that are the typical ones that are icosahedric and material, and the lytic effect on a culture spread of Xylella. A long list, they have a very long list of bacteriophages, and these bacteriophages are submitted to the same process of screening in the previous presentation. In planta, okay, and also in the field. So, these are the deliverables we had from this work package. Consortium of 5 strains, botanical extracts from alperujo and from the eucalyptus oil, 2 peptides. This is one peptide which is the consensus sequence from a peptide which is endogenous in all Rosaceous plants. I'm not going to detail, but it was obtained from sequencing from genomes and finally doing a consensus sequence. And this consensus sequence, which is more prone to prunus, was effectively reducing defenses and providing priming to the plant, to almond. And this is the bifunctional peptide. And finally, the bacteriophages. So, the final message, because across the presentations of the project since the first stages, there are many questions. People say, okay, you have provided new solutions, but when the grower will have these solutions on hand? First, more field testing for validation. This is clear. In work package 7, we are ongoing on that, but believe me, it's not sufficient. We have to develop formulations and application systems, more compatible, obviously, with the agricultural practices. Also, endotherapy is a growing methodology now in several diseases. Apply for registration. In Europe, this is a requirement for any substance or microorganism under the EU regulation. Preparation of the dossier for the registration. This is based on active substance, and both are necessarily produced by companies, because the money is on the hands of companies, and companies have the interest in commercializing these products. But finally, production and formulation. All the technologies can be produced and formulated. This is clear in one or another approach. And finally, authorization of a product based on the active substance in every country. This is the regulation actually in Europe. Finally, commercial delivery to growers. So this means that being very optimistic, minimum 5 years. Of the technologies I have presented, probably bacteriophages is the most close. Then the consortium. But in the case of the consortium, the problem is you have to register every strain. Okay, so more money. Not now, but to register it as a plant protection product. You can register it as a biostimulant or you can register it as a biofertilizer.
The data requirements for microorganisms will change, so you're saying, but you can't register consortium.
Okay,
But it's a choice, but there is a possibility.
but you have to provide
Data for a consortium,
for everyone.
for a consortium, yes.
Only for the consortium or also in the dossier, you have to provide information on every strain. Okay, okay, but you can register it.
It's one dossier, no?
Okay, it's one dossier.
Mhm.
Okay. And peptides depends on the peptide. There are peptides, for example, the most promising are the plant defense stimulating peptides, which are priming the plant, which are active at very low concentrations. This is important. And in many cases, do not need to be inoculated by endotherapy. It's enough to spray the plant. And that's all. Many thanks. Sorry.
We have some minutes for questions, so we're proceeding with the last two for packages. No question?
Yes, thank you for the presentation. I wonder, you are already working with some companies in the development of these technologies or these solutions. Or now
What-
we need to.
Yeah, that's yeah. You're right. It depends on the product. For example, for the eucalyptus oil, there is a company these results are part of the work with the company, but is also on the project. So this is a way of saying to a company, why not to test this product for Xylella? You say, wow, it's the problem with the companies is that they have to see a market. And Xylella, for some companies, is not still a market. Yes, that's the real world. So to develop a product, they have to gain benefits, and that's the problem. For peptides, we have been working with two companies in the past, not for Xylella, for other applications. And the problem is that they were not very big companies. These companies were bought by big ones, and the big ones only see sales and benefits. And they say, wow, this is very interesting, but we don't go further. The peptides, the main problem is the cost of synthesis, which is not very well developed. And in some cases, for example, one peptide, which is insecticidal, is at this time under revision in the EFSA. It has been produced by yeast. So it's using a yeast platform for producing the peptide. The peptide is not considered transgenic. The yeast, yes. So well, this is a way of decreasing cost in many cases. But yes, and we are still working with trying to convince companies, big companies. For example, Bayer, we have been talking with Bayer, but since Bayer was joined with Monsanto, or Monsanto was to Bayer, we don't know what. Finally, they abandoned the idea, but we are fighting on that. And the viruses is the company.
Yeah, Evolving Therapeutics, that is a spin-off of the University of Valencia, Associates. So they are working on the production of bacteriophages and doing the tests in the field. So for the endotherapy, we have some collaborators. It's a company specialized in developing devices. So the company has provided the endotherapy devices for testing under field conditions. So they were quite interested. So we have had to apply the products in the plant, but what we need is a company that is.
They don't have your name, but if you read the paper from Marcos Corpicini, you will see that. Innovation, the treatment, the way of application of the treatment, and the technologies are emerging technologies. I think we have very good future, but need more implementation and more action to convince companies to arrive to growers, but they are very promising. Really.
Maybe we can move to work purpose.
Good morning, everybody. I'm Gianni Giglioli. I'm professor at the University of Brescia, Italy. And I lead, together with Diego Olmo, the work package 7 tools supporting the design and implementation of tactics and strategies to mitigate the impact of Xylella fastidiosa. The aim of this working group is basically to test and validate available technologies in order to also innovative solution to mitigate the impact of Xylella fastidiosa. And second, to test if we are at the stage we can put together all the available solutions to define schemes for going in the direction of integrated pest management. And these are the 12 participants in this work package. And I try to be quite simple in terms of communicating the main aspect of each task, starting for the first one that is devoted to the development of integrating modeling framework for exploring epidemiological scenarios. So, and models are tools, as anticipated by BeXyl, that could help in understanding not only the system, but also in guiding the management of the system. For that, we start at the beginning of this project, two main activities. One is devoted to the simulation of the vector dynamics. And for that, we develop a phenomenological model based on what is known on the Phineas vector, trying to group all the knowledge available and evaluate data collected in Apulia and also in other places in Italy. And we obtain a good model precisely representing the phenomenology layer of all the stages. And the second one is a demographic model, adding the abundance, not only the phenomenology. This is an improvement on one side. On the other side, you are more exposed to variability and uncertainty regarding the definition of initial condition that could transfer uncertainty to all the forecast in terms of the dynamics. In any case, the results are quite good based on my experience. And we expect that these two tools could be useful for defining the best timing for monitoring and control. They are ready. This is important, and not only scientific output, but really a tool for supporting the management. Then we work on epidemiological models. An epidemiological model, the one that the tool that we have developed, are complex, putting together all the elements, host, cultivated plants, natural host, vector, the progress of the disease within a plant, and climatic, local factors, also arrangement and time spent by the vector in the different compartments. We obtain quite good results in terms of the description of the epidemiology noise and relatively preliminary but satisfactory results in grapevines. The usefulness of this tool is again to support risk assessments on one side, but also explore the efficacy of IPM strategies. Then there is task 7.2, best agronomic practices for the management of tolerant and resistant cultivars in affected areas. The purpose is to design an experiment in which different factors are put together, the type of cultivar to Fabulosa Vecina that are resistant and tolerant, the management practices in terms of soil tillage and mower mulching, and the density of the planting, Fabulosa super high density or high density, and Vecina low density and high density. There's also another variable that cannot be controlled, so the vector density that is very much important in terms of pushing the disease. The response variable were prevalence and within canopy infection dynamics, because we observed in this task also the dynamics within the plant. So the results are that the progression in Fabulosa cultivar is higher, so the rate of increase is higher, was clearly observed with respect of Vecino. Although there is an uncertainty related to the appearance of the symptoms. What was more probably surprising is the variability in appearance of the symptoms, but also in the capacity of detecting the disease within the canopy. These are very important data, not only for validating the model, but putting more knowledge in terms of the dynamics. We don't know enough regarding the real temporal dynamic of the disease. And this is a piece of knowledge that is very much required for understanding the epidemiology of the system. Then the testing solution, I think that we can skip it because you have already presented.
Apologize for spoiling, but
yes, you have. So we part of the presentation.
But this is a true integration between two different working packages.
Time. Thank you very much. So I skip everything, and there is the also was anticipated before the fact that in the task 7.4. We are developing a network of plots for monitoring and demonstrating innovative tools for the management of Xylella fastidiosa. It was already anticipated how it's complex, this process, and how that it has probably last years to understand the efficacy and the dynamics of the disease in treated plants. These are the places in which we are implementing these new technologies, peptides in almonds, phage cocktails in grapevine, and zinc cones in olives. There are 5 testing places in Mallorca and 2 in the Salento Peninsula in southern Italy. So it's a long-term assessment. Again, it's a matter of waiting appearance and symptoms of the symptoms and testing plans in order to understand what really is the dynamics and the efficacy of these new techniques. Having in mind the different level of TRL of the technology in terms of possible application. I come back to this point in the last task. These are the places where the trial fields are located. And then the last one. I dedicate more time on that because it's an ongoing activity. It's an important activity. It's not finished, so I only anticipate probably more methodological issue than results. If you are able to come to Apulia in June, or at least to follow by web the conference, result will be presented, I hope, with the help of Blanca, that is pushing a lot. Our expert, asking them give an opinion. So I'm joking, okay, but it's important. I'm joking, but it's true. Okay, so I start from an important aspect. So Xylella fastidiosa is also related to the economic aspects. No, it is a quarantine pest. It is regulated, and the management is completely different. What is the current management for farmers that is based on IPM? We expect really an economic improvement, all the management options or feasibility, only if we are able to go in the direction of IPM. We are in between. But in any case, we have to tackle this phase in terms of transition from emergency action to sustainable management. And this is because we dedicated a lot of effort in developing a decision support system, a DSS, trying to define a preliminary set of IPM strategies under endemic scenario. So it's not possible to talk about IPM strategies in a different place apart from endemic location. Okay, what is IPM? I think it's not necessary to explain, but I would like to dedicate small attention on the definition of IPM option for us. An IPM option is an intervention aiming to reduce the population abundance, all the population in the epidemiological systems. And they are organized in three phases: prevention, monitoring, and control, according to the classical way for an IPM. We have defined in the project an IPM scheme, a coherent and hierarchical organized set of IPM options. Of course, we put together. By putting together, we can reach any kind of complexity. So not only, but we have to look at the context in which we want to implement IPM. IPM is not the same everywhere. It has to be context dependent. So to look at the context, we have to take into consideration legislation constraints, the crop type, the local management practices, the actors involved, and the socio-economic aspects. I come back to it later. So we started an exercise. It is a structured expert judgment collection based on different phases. I don't want to insist on the methodological aspect, but together this structured expert judgment collection, there is another stage of the work that is performed at work package 8. It is the stakeholder interviews. So in this way, we are able to go in the two directions. One is the more technical one, and this in terms of judging the effectiveness, the level of knowledge, the potential impact in environmental and human health impacts. But not only, we are able to evaluate the acceptability regarding also the readiness of the adoption of the schemes based on considering the economic cost, the acceptability, the ease of implementation, and the availability of these options. So, what is the result? The DSS platform has been designed considering the different actors. And this is the first level: farmers, nursery, risk managers, and then looking at different levels of information, the crop types, the species, almond, grape, olive, the objective of the actors. We have classified it in two main categories: replace or start new plantation, or manage the current crop. Most of the cases, we expect this is more important, but in any case, also the possibility of introducing resistant cultivars is one of the options to be considered. Of course, it costs more. Another important, so you probably had a spine in my university because you already mentioned the different level of intensity. So
I simply contribute to the evaluation of
you are
right
one of the no, it's a matter of, okay, you can design any kind of scheme. Of course, this as an outcome, the different level of intensity, low, medium, or high, that correspond to high capacity of control, but higher costs. It's a matter of balancing the decision taken by the user, the end user, where he or she wants to go. We describe in this way different schemes. We have developed many schemes. They are commented, I would say, so analyzing the feasibility according to the context dependence element. For example, if it is important to irrigate, but you are in a place where you don't have enough water, or you manage an olive crop without any contribution of irrigation, doesn't make sense. So it could be a very good scheme without any applicability in the context. So, and then we go in the direction of implementing. So we are working in the software, also the interface. This is a preliminary, everything is preliminary now. And you start defining who are you. You select farmer, nursery, or it is not represented here, the risk manager we are discussing, if it is the case that we involve it, this category. Then you define the crop in which you are interested. We have a second step. And at the third step, you define your objective. And then probably the most important one, together with the consideration of the context dependence element, the intensity level. This is a decision. You go in a certain direction, you obtain a certain result, but of course you can back and explore different schemes, no? And this is just an example. So at the end, the scheme is represented by a set of measures regarding the prevention, the monitoring, and the management, the intervention. So the practice that could imply an intervention on the populations. What we have done a set of interviews, or better, questionnaire that was sent to the expert. We collected their opinion for all the schemes that we have developed, organized in terms of level, in terms of all the information layers that I showed before. And we have grouped the answers considering two main elements. One is a judgment with a score from 1 to 10 regarding the integrated performance. And this comes from the experts on the disease, but also risk managers. And the other one that comes from the stakeholders is a measure of the adoption, a judgment on the adoption readiness. And then we are now also considering an opportunity to include the confidence level. That is a measure of the variability in the answers. This is just a measure of intensity, could be the mean or the median, okay? And this is an expression of how confident are the experts in judging this specific scheme. So this is a good scheme with low uncertainty. You see, this is a good adoption level with levels of uncertainty that in this case is higher than in that case. Okay, I think that, no. Then this DSS contains important information. We have developed leaflets illustrating on a certain level of details all these schemes. Then each option of each scheme is described technically at two levels, general guidelines regarding the option, and the second one in which we provide more specific and detailed guidelines. So, and you see there is also online materials to support the readers and the user to have a more deep analysis regarding the information that we provide. We hope that by the end of June, the platform will be ready, hosted in, I don't know exactly where, because probably we need to have a server that is not in our university, is never in the project, but is a technical detail. I will give at that time the web address to test it, to look at it, and I hope also to give your feedback. Thank you.
I'm used to it.
But not like the best.
Exactly, the best by far. Yeah. Okay, good afternoon to everyone. So somehow socio-economic assessments always come in multidisciplinary projects at the end, and work package 8 dealt with the ecological, social, and economic impacts of Xylella fastidiosa outbreaks, but also of the management activities. This work package had 14 partners out of the 28, led by Forest Research in the UK and the Joint Research Centre in Seville. And I will mention which are the lead partners in each of the activities. So basically, we had 4 main areas of analysis. First, we want to review how Xylella fastidiosa has been managed since its start in 2013 in Italy and 2019 in Alicante. Then we wanted to know what stakeholders actually know about Xylella and what are their attitudes about the management options. So Gianni just mentioned that this readiness level. So we are trying to look about this readiness level. Then our colleagues from the University of Córdoba and CSIC also wanted to look what are the ecological impacts, not only of Xylella outbreaks, but also about their management. Yeah? So we have a Xylella outbreak, then we want to see what happens with soil characteristics, biological communities, and things that I failed to understand very well, but Pablo gave me some graphs that I can show to you. And last, we wanted to see also what are the economic impacts of Xylella fastidiosa outbreaks at a global level and on the management at the farm level. So regarding the first activity that was led by the Consiglio Nazionale della Ricerca in Bari, Pier Federico Lanotte and his colleagues. So what they've done is they've done 3 main paths. The first one, they've done an expert group with the food and veterinary offices in France, Portugal, Italy, and Spain, and try to understand what kind of strategies they need to manage the outbreak. This was based with a standardized reporting template that would allow comparing the strategies. So we have a common strategy that is the Commission regulation, but then that it's implemented differently in the different areas where it happens. In Spain, for example, at the level of the regional authority and so on. And we wanted to know exactly how these strategies were designed and how they are being implemented. Second, we wanted also to have a quantitative overview of what has been done in terms of type of actions, the intensity, in terms of number of times they have been done, and in budget, how much money has actually been spent to control Xylella. There's also an analysis of the representation of Xylella fastidiosa related news in the media to try to understand the role of misinformation in creating resistance and conflict between managing authorities and farming communities. And there's also a questionnaire to stakeholders in Italy, Spain, France, and Portugal regarding how they have experienced these control measures. This is still open, so reach out if you still want to respond. The questionnaires are online, and Federico's team is currently analyzing them. The second activity, which is led by Forest Research in the UK, is to understand what stakeholders know about Xylella. How does the type of situation in the region affect the learning process about the bacteria, and how does also the experience with Xylella change the way they want to tackle the bacteria. So here we are lucky because we have, well, we're lucky or unlucky, because we have three very specific cases, both in terms of the legal status. So we have an eradication area in Alicante, we have a containment area in Italy, and we have a monitoring area, which is basically an area at risk where we still do not have not found Xylella. And so we wanted to see how the narrative about managing Xylella changes as the experience with Xylella happens. These were based mostly on interviews and focus groups with over nearly 150 farmers and their representatives, approximately 50 each. What we see is that there's three very distinctive perspectives. So from the worst scenario, where Xylella has been there for more than a decade, to the best scenario, where Xylella is expected or a threat. So what we find in Salento is that basically Xylella is no longer a discussion item. The discussion focuses on what do we do after Xylella. So Salento is BeXyl. It's beyond Xylella. What is the future? They really want alternatives. They are desperate for something that can allow them to keep on farming in the area. And we will see that in the fourth task, where, contrary to our expectations, farmers do not need to be compensated to test alternatives. They are willing to pay for alternatives, which is quite surprising. There's also a high level of trust in science, but they are tired of waiting. They really trust science, but patience plays a role. I mean, they've been promising solutions for far too long, and nothing is working. In Alicante, we are 5 years in the past compared to Italy. So basically, Alicante is conflict. I mean, Vicente is not here, but Vicente can tell you what conflict means when Xylella appears. They are really tired of eradication. They assume that eradication is impossible, and they don't understand why they do not move to containment, where they can start living with Xylella. They don't see future for their activity under an eradication plan, and they are afraid of Xylella jumping to additional crops. It's now in almond because they have fastidiosa. Multiplex, and that's how they might arrive, and also not allow them to grow olives anymore. And then in Cordoba, I mean, we all believe, or we all think that Xylella is a well-known topic by everyone, but there's a lot of ignorance. They've heard about it, but they really do not know what it means. They have little trust on what might happen when Xylella appears in Andalusia, mostly because of what they hear from the colleagues in Alicante, and the awareness of the risk is not very high, which is quite funny. And then when we test how they look into the prevention strategy of the IPM, so not the schemes, but the options, what we see is that there's a consistent preference for anything that allows them to be Xylella free without changing the way they farm. So So if they have a resistant multivar, if they have a vaccine, these peptides that Emilio has mentioned, that they are willing to do. What they are not so willing to do is this idea of changing the way they farm. And also, there's an unanimous view that there's a need for coordination action, that whatever they do alone is not gonna help them. And we see that the acceptance of the blue, which is basically the prevention, is quite similar. The monitoring is more accepted in Alicante and Salento and not in Córdoba because they really do not understand what they are looking for. And then what we see is that the intervention based on eradication is now given for granted in Salento. And the further you move away from Xylella existing, the less acceptance there is. The third aspect that we have looked into is the ecological impacts of outbreaks and management. This has been done by the University of Córdoba and the CSIC in Seville. And basically, they have been checking 19 plots in Alicante to see how the diversity of soils and biodiversity, change between non-infected, infected, and treated. And what we see is that there's a direct correlation between the phytosanitary status of almond trees and Xylella fastidiosa. However, the environmental conditions affect this relation, and in the natural vegetation, Xylella fastidiosa does not seem to affect this phytosanitary status. So it really affects when we are looking at the host plant, but it doesn't affect so much to the scrubs and the CTI that I don't really know. This is well beyond my comfort zone. And then when we are testing the green, that is the Xylella free, the red, that is the Xylella infected, and the yellow, that is the Xylella eradicated, we see first that when there's an infection, the bacterial populations fall, but after eradication, they revert to the original level. The bacterial diversity falls, and that is not recuperated after eradication. So you see in the bottom left, the red and the orange bars are not significantly different. They have the same letter above it. And the same happens to the bacterial richness. So eradication increases, reverts the population to the baseline level, but does not revert the diversity or the richness. And regarding the physicochemical properties of the soil, we see that eradication allows returning to the baseline levels of total carbon, but not so much for calcium. So eradication may have unintended consequences on soil health and microbial resilience, and there might be a need to revisit the way we are eradicating Xylella to make sure that it doesn't have unintended consequences in the ecological diversity of the affected areas. And the second thing they did is they revisited the risk of Xylella in Spain. So basically, with information on the potential hosts and the potential vectors, how they are distributed, together with the habitat suitability from Ponte, I believe, which is the Schneider et al. 2020 paper in the Proceedings of National Academy of Science, they include another layer, which is the exposure to the introduction risk based on local, where import places are located in Spain and also nurseries, which I guess these are the nurseries and these are the import places. We end up with the risk map of Xylella in Spain that basically is quite strong in the outer areas of the peninsula where there's straight exposure and good climatic conditions, not so much in the north, and it decreases as we move inland in Spain. So I assume that this risk map is under review or about to be published by our colleagues in the University of Córdoba. And the last thing that we have done is to check the economic impacts of Xylella fastidiosa and outbreaks and the management. This is what I've been leading, so I'm much more comfortable telling you about this. And we have done basically two approaches. One is a global approach to understand what Xylella might actually mean beyond the 5.5 billion euros at risk that were reported in the I2P2 indicator for the priority pest ranking. We want to see how that is distributed and how that changes with IPM schemes. When we have used the IPM schemes, we have followed a bit the logic of Yanni's presentation. We have a low intensity IPM and a high intensity IPM. However, due to the nature of the global scale analysis that we are doing, we have to really simplify into the two driving parameters that affect markets. One, what happens when Xylella is there? So when Xylella is there, we assume the yield losses of EFSA's assessment of Xylella for the priority test. And then a moderate IPM scenario manages to reduce the yield losses by 50%, and an intensive IPM scenario manages to reduce yield losses by 75%. And of course, the more intensive the IPM management is, the more expensive it is. Yeah. So basically, the intensive IPM scenario is 2 times as expensive as the moderate scenario, and increases 50% the effectiveness in control. So we are doing it for almond, citrus, and olives, and I'm going to focus on the results for olives. So basically, on the top, we see what are the yield shocks that are reported by EFSA, and by definition, when we don't have IPM, they are quite high. I mean, they are between 50 and 70%. IPM, the moderate effect, reduces them by 50%, and the intensive by 75%. Once we introduce those shocks into the model, we see that there's a reduction on the area of olive oil, of olive trees. I mean, they are eradicated, they are taken away, and something else is done because it's no longer profitable. And there's some areas where the factor increases, mainly because they have a very small area in the baseline. And the reduction on olive oil area also decreases with IPM implementation because the yield losses are lower, so they are remaining profitable. The fact that we have a reduction on area and a reduction on yield means that we have a much lower supply of olives for olive oil in Europe if we have an expansion of Xylella fastidiosa, and this expansion is reduced the more we manage it. And the lower supply leads to higher prices. So the prices increase marginally with the reduction on production. Combining the fact that we have a quantity effect reduction and a price effect increase, what we see is that the quantity losses are much bigger than the price effects. And the income, the revenue of olive growing in Europe will reduce if we don't control Xylella. And it will reduce quite dramatically. Even in the best case scenario, we have nearly a 15% reduction. So, I mean, Xylella, if it expands, will still affect no matter what we are doing to control it. And even if the revenues of the sale of olives decrease, in some areas, that is compensated because of the savings that they can attain. Because of the increase in the production costs is not so big. So it's a bit more diverse, especially Sardinia, and I think that is Tuscany there. The report explains a bit what is the underlying factors for this diversity. Well, that is mostly the same color in every place. When we are talking about income, there's more diversity in colors, mostly on the reds, and with those 2 green areas. And basically, the message from this aggregated analysis is that IPM pays off. We've been always saying that IPM is great, that we don't understand why farmers don't do it. So this de facto says, so in from the no intervention to the moderate IPM, we see that we are saving 2.5 billions, so a 58% increase over the baseline. And if we move from the moderate to the intensive, it's 3.8 billions. And de facto, what we see, a nice figure that you can quote, is that if we implement IPM to control Xylella, we are de facto assuring 3.9 billions of production in olives in Europe. The second part relates more now to specific farmer behavior, and it links to what Yanni has been saying. What he calls options, we call attributes. Yeah? So control the vector, increase plant resilience, use less pesticides, and what are we paying? And what he calls replace or manage in his decision support system, we call vector control, plant, or plant and vector. So we are trying to understand what farmers want. Do they want to replace the hosts, as we saw in the qualitative inputs from activity 2, or do they want a more integrated approach? And here we have run interviews with 600 farmers, so we really increase the scope at the cost of decreasing the depth of what we do with farmers. For olives in Puglia and for almonds in Alicante, and from the analysis, we will be able to understand which is the strategy that farmers accept more. So in the decision support system of Yanni, how many people will click on plant, or how many people will click on vector, and we will also understand how much money do we need to pay farmers to actually implement this option. And we will know this for individual options and also for combinations of options or schemes. The second part we did is we also want to know how can we change the eradication policy once it's detected. And here we are focusing also on developments of other work packages, and it relates to early detection asymptomatic technology. Currently, you detect a plant and you tell the farmer that the plant is sick, the plant is dying, there's a radius that you clear, there's a level of a set of hosts that you cannot plant anymore. So we are trying to understand if they would accept that there is detection and eradication taken without this confirmation by PCR. And here what we are considering is how much they are willing to take preventive action under uncertainty, because of course the accuracy rate of the new technologies, of these early detection technologies, are not as high, 99%, as the PCR in a lab. We want to see if by detecting before, the fact that the radius of eradication could be smaller, because in theory the bacteria is not so prevalent in the sap of the tree, and the beetle bugs might not have taken it and started spreading it. So maybe we could play with the eradication radius, and also about the amount of money that we need to compensate farmers to accept first the higher uncertainty regarding the fact that we might have made a mistake, and the fact that they have to believe that the tree is sick despite that he's not seeing the tree sick. Yeah? So we want to know if these technologies would be acceptable for farmers as detection, and what is the additional money we need to go, and basically play with the radius and the compensation. We could eventually have a cheaper eradication technology. We might need to pay more, but for less square meters. Yeah? And So the last thing is, okay, we want to know what farmers want, but society includes both farmers and citizens, and we want to know what kind of restoration landscape is preferred by farmers. Here we have worked in Italy and Spain. We have 2,000 responses that we have obtained. And for example, the first difference that we see is that indeed Xylella is something that people talk about in cafes and bars in Italy, but not so much in Spain. I mean, it really shifts down. And what we are working on is what kind of agricultural system we're going to put after Xylella. I mean, is it going to be super intensive production, abandoned fields, which is currently what's happening, or are we going to do a mixed crop production? What kind of Xylella control measures do we want? Do we want high-tech, fancy things like peptides and things like that, or do we want low technology control as the ones that Bosco presented? Yeah. So these things that repel and attract the nymphs, or basically insecticides and tillage, which is what's currently done. We want to know who is funding, who's managing the funds, and how much actually society is willing to pay. So we have found that there's a clear preference for mixed crop restoration. People will still want some kind of traditional landscape where Xylella has been found, and that is about 31 euros per household and year. They really want high tech, so that they believe more on fancy high tech solutions than on classic green cover management. And there's some preference that the funds are managed by the farmers themselves and not so much by the regional or the national government. And this is what we've learned. We come last, not only in the presentation, but also on the work, because we had to wait for our colleagues to develop the solutions that we had to test. And we hope, well, Anna and Blanca hope that the 31st of July our development. Thank you very much.
So time for questions before we go for lunch.
Yeah. I have a understanding question, maybe I missed it. It's very interesting decision scheme platform. Do you want a lot of detail of which parameters or products you use?
Yeah.
So it's not at that level.
Maybe can you mention
very
a
tricky,
bit that's listed, but not at the level of the activity or they're more the evolution of the information.
It would be very tricky to have it at that level because it's sustainable starts at certain dynamics.
Isn't that we have to do it outside the market?
Yes, and it depends on the member space for things. Okay.
You know, if you do the detached joint, your reason, but also the market reason, and also the variability of the comments. It is already considered, but not as a shell.
Or maybe a comment rather than a question, which is kind of philosophical, intrinsically contained in the title of the project. This was beyond Xylella. So we discussed of several possible solutions that could be implemented, but just to avoid misunderstanding, at least for my part, but I think for all of the contributors, we are trying to provide new solutions. This doesn't mean that solutions that have been implemented and forth so far are bad solutions. For example, it's really my opinion that we cannot continue on the long run with plugging, for example.
but this doesn't mean that I'm against implementing this measure in this phase. And also, okay, this message is something like, I appreciated the message done by Emilio that said, we provided proof of concept. This is not the product that will be in the market tomorrow. So just to avoid misunderstanding, I think we worked hard these years. We can provide, we suggest new solutions, and that can, I hope that some of these can be implemented even in the next future, but of course, this does not mean that we are against the measures that have been implemented. Even the chemical insecticide, for example, of course, we did not devote any effort in this direction because those are already available. But I'm not saying stop absolutely with insecticide. We know it's not sustainable in the long run, but that's all.
Okay. So thank you again for those that came today, and also for people that are connected online. I'm sorry for you because you are going to miss the paella. And now I hope we can have some time and also talk a little bit. Okay, thank you.